So this is a part two of our hemoflagellates lecture. So we will be discussing more about the Trypanosoma species. We're already done of your Leishmania species. So let's go with your Trypanosoma. Under Trypanosoma genera, we have two important genus and species, clinically significant genus and species, Trypanosoma cruzi, Trypanosoma brucei complex. So trypanosomes in general are subdivided into human trypanosomes and animal trypanosomes. So in here, we have Trypanosoma cruzi, Trypanosoma brucei, and Trypanosoma wrangeli. So for animal trypanosomes, we have Trypanosoma brucei, Trypanosoma evansi, Trypanosoma lewisi, and Trypanosoma equiperdum. So for the clinically significant parasites, we will just be talking and focusing our lecture to Trypanosoma cruzi and Trypanosoma brucei complex. So in here, just like your Leishmania, trypanosomes also show a very strict geographical distributions. So these microorganisms or parasites in general are actually found in the South American region. That is why most of these parasites are transmitted and could generally cause African trypanosomiasis. So African trypanosomiasis, which we will be talking more about that later on. So let's start with Trypanosoma cruzi. So Trypanosoma cruzi, just like your Leishmania species, it is also a zoonotic disease. Therefore, it also involves two different hosts. So we have the vertebrate host, which is the definitive host. And we have also the insect vector, which is the redovid bug. So these are the redovid bugs or the insect vectors are considered as the intermediate host. Take note again the difference between these two hosts. When we say definitive hosts, these are the hosts that acquire the sexual multiplication or reproduction of the parasite. When we say intermediate host, this is the host wherein the parasite multiplies asexually. So in vertebrate host, we have two forms. So we have the trypomastogote and then the amastogote form. So for the insect vector or our intermediate host, we have all four forms. We have the trypomastogote, amastogote, promastogote, and then the epimastogote. Therefore, class, the life cycle of Trypanosoma, specifically your Trypanosoma cruzi, is much more complex compared to your Leishmania, since all of the four morphological forms of this parasite exist and are intermediate host. So since it's a zoonotic disease, as I have mentioned, these parasites also have animal reservoir, just like your Leishmania. So these animal reservoir ranges from dogs, cats, until rodents. So let's focus more about the basic morphology of Trypanosoma cruzi. So we have here the trypomastogote form. I think we have already mentioned, I have already introduced this one to you on our the first few slides on our lecture under Leishmania. So for the trypomastogote form, just remember that your trypomastogote form exists in a spindle shape. So it appears as C or U shape, or again, in some books we have the S shape. So the trypomastogote form is subdivided into two forms. We have the long and slender form, and we have the short and stubby form. So these trypomastogotes are usually seen in the peripheral blood smears, but it it shares the same morphology. So still it consists of a central nucleus, as you can see here, a central nucleus and a large kinetoplast situated posteriorly of the parasite. So where's the kinetoplast here? This one. So, so from the kinetoplast, flagellum originates and it actually transverse throughout the body. So this is your flagellum. This is your flagellum. This one. So it transverses throughout the body, forming an undulating membrane. So let's focus here to this image. You could actually see the undulating membrane. This one, that is the undulating membrane. So it is actually found in the anterior end as the free flagellum. So we have here the elongated nucleus. Again, the subterminal kinetoplast and a short free flagellum. Take note that tripomastigote forms are the invasive forms. They are the invasive forms, meaning they have the capacity to migrate to many organs. So have the capacity to migrate to many organs and penetrate these organs and these cells. That continues the life cycle of the parasite. So this this broader form, or the short and stubby form, or usually has longer flagellum. It has longer flagellum, but they are less motile, so compared to the long and slender forms. So in here you could, for this photos, you could see actual microscopic examinations from figure A. So we have here trypomastoid in blood smears stained with still with gemsa. Figure B is just a higher magnification of figure A. Figure C is figure C is still a trypomastoid stained with, but this is a thin blood smear compared to the first one, thin blood smear. So we can see in figure C more anterior location of the nucleus. So the nucleus is found in the anterior portion. We'll show you another picture right here, a figure. So in figure A, you could appreciate this is still a thin blood smear stained with gemsa. You could note the typical C shape of the trypomastoid. So this characterizes your T. cruzi. So T. cruzi. So we have here the C-shaped trypomastoid. Trypomastoid. So we could see here typical C shape of the trypomastoid. And figure B, we have here three trypomastoid in thin blood smears. We have one, two, three. Figure C, we still have here trypomastoid still with thin blood smear. In here, you could appreciate the undulating membrane, that one. Still trypomastoid. So another morphology or stage of your urticacy is the amastigote form. We already know about amastigote. This is the non-motile form, so it is usually round to oval. It has large nucleus. It has kinetoplast, but it has no flagella. Again, this is non-motile. Usually, again, amastigote form is the tissue stage of the parasite, meaning they are found inside the tissues or in the cells. If your trypomastoid is the non-multiplying form, your amastigote is the multiplying form, just like in this pictures. So what is depicted in this picture are actual microscopic examination of Trypanosoma cruzi, a mastigote in the heart tissue. So this is the heart tissue. So inside in your heart tissue, this one, that one is your amastigotes. So this is actually stained using H&E. This is stained using H&E. Figure A and B are the same. Figure B is just the magnified version of figure A. So just take note, when fully developed, a large number of amastigote may be found in a cyst or a like cavity. So this is indistinguishable from those found in Leishmania infection. So this is indistinguishable. So your T. cruzi passes its life cycle still in two hosts. So we have a complicated life cycle. So we have humans as the definitive host. We have the vector or the redovid bugs, which is the intermediate host. Redovid bugs or the kissing bugs or the triatomin bugs. So these are the same. So these are considered the vectors or the intermediate host of the parasite. So the infective form of the parasite or the infective stage of the parasite to humans are still metacyclic trypomastigote. It's the metacyclic trypomastigote. Again, please take note because for your leishmania, it has a different infective stage. So for your Trypanosoma cruzi, the infective stage to humans are metacyclic trypomastigote. So the metacyclic trypomastigote form, it is usually found in the feces of the redovid bugs. It is found in the feces of the redovid bugs. Why? Because the redovid bugs, after feeding, they usually defecates. So not necessarily that this stage of the parasite is actually found in the gut, inside the gut of the parasite. No, so it will not migrate to the salivary glands, transmitting the form, the stage to the skin abrasions or to the directly inoculating it to the skin. So what the bug will do, it will feed to the host, to the human host. After feeding, it would defecate. And since there is already a lesion, so there already a punctured wound, so an exposure of the feces may cause the patient be infected with Trypanosoma cruzi. Again, after feeding, the bug will defecate. So the exposure of the redovid's feces may acquire or may could cause the patient of the acquisition of the infection or of the parasite. So these redovid bugs, just take note, are nocturnals. So they actually feed during nighttime. That is why Trypanosoma cruzi are known to cause sleeping sickness because they feed on humans while the humans are sleeping. So these humans get infections again when their skins are abraded, so or if their eyes are contaminated with the feces of the redovid bugs. So these are some of the basic or most common transmission of Trypanosoma cruzi. But aside from that, Trypanosoma cruzi can also be transmitted by or through blood transfusion. So through blood transfusion or in some severe cases, organ transplantation from mother to fetus. So we could also uh, entertain the idea of congenital transmission, although very rarely, it still can happen. So some 5 to 10% rare cases could occur from congenital transmission and also contaminated food and drinks. So just take note that the feces of the redovid bug harbors the infective stage of the host. So again, the feces of the redovid bug harbors the infective stage, which is the metacyclic trypomasticote. Take note that the redovid bug doesn't have metacyclic trypomasticote on its salivary glands. So why it is still correlated with the feeding or the biting of redovid bugs, so upon biting, there is an abraded skin. So there's a punctured wound. So there is a very big possibility that the feces of the redovid bug will be contaminated to the or would contaminate the abraded skin. And that is how the humans could actually acquire the infection. So aside from that, very important as medtechs, we should also know that we could actually get this through laboratory accidents as well. So through maybe needle prick injury, accidental needle pricks. So that is actually how we get this as well. So this is the life cycle of Trypanosoma cruzi. So just like your Leishmania, we have two different life cycles. So first is from the mammalian stages or the definitive host, and that is the humans, and we have also the intermediate host, and that is for the vectors. So for the Trypanosoma cruzi again, we humans are infected with the feces of the redovate bugs. So containing the metacyclic trypomastigotes. So in here, so triatamine bug takes a blood meal. So right after taking the blood meal, usually the bug again defecates. So that is wherein the mucosal membranes get contaminated with the infective stage, which is the metacyclic trypomastigotes. So the metacyclic trypomastigotes will be inoculated in the bite site. So from metacyclic trypomastigotes, it will become a mastigotes. So it would invade the reticuloendothelial system. So still, it would invade the reticuloendothelial system. So we're still talking about the spleen, the bone marrow. So those are still the same organs that these parasite could invade. Again, for hemoflagellates, their very unique characteristics are they have the capacity to invade tissues and cells. They are obligate intracellular parasites, so meaning they live inside the cells. And number three, they attack or invade the reticuloendothelial systems or the macrophages in general. So from metacyclic trypomastigote, it would transform to become a mastigote stage. So it would still multiply by binary fission, and it would actually become a pseudocyst. So right here, hindi na no. So from here, it actually still perform. It will multiply to become a pseudocyst. This is a pseudocyst. Actually, this one. This is the presentation of a pseudocyst. So inside the pseudocyst is the motile, C-shaped, but non-multiplying form. So when the pseudocyst ruptures, so the intracellular amastigotes will transform to become a trypomastigotes. So they will burst and they will invade the bloodstream. So again, uh, the bloodstream is the highway inside our body. So if any organism that has the capacity to invade the bloodstream, it has the capacity to invade distal organs, most important distal organs, and that includes maybe the heart, that could include the liver, the spleen, and so on. So this actually, so when they would become uh, invade they will when they invade the bloodstream they could actually go to the other organs or in the if you could remember, the trypomastigotes are subdivided into two forms, right? We have the high motile and then the last motile. So the slender or the high motile form of trypomastigotes is the culprit of invasion. So they will invade to the migrating organs, while the broader form of trypomastigotes will stay or remain in the blood side. So they will remain in the blood side. So right here, it is here. It is actually demonstrated in this life cycle. So it will transform, the amastigote will transform into the trypomastigotes and it will enter the bloodstream. So this is actually the broader trypomastigotes. So these broader trypomastigotes are less motile, if you could remember. So they are less invasive. So they will remain in the bloodstream. While the other trypomastigotes, specifically the slender trypomastigotes, has the capacity to invade. Why? Because they are much motile. They are more motile or high motile. So they will move, migrate to other organs, and form another infection sites inside the body. So that is the invasive form of the trypomastigotes. So in here, when the broader form of trypomastigotes is still found in the bloodstream, that is the same stage where the which the triatomine bug or the vector takes. So therefore, the trypomastigotes is the infective stage of the vector or of the intermediate host. So your trypomastigote stage is the infective stage of the intermediate host. Remember your trypomastigote stage, specifically the broader trypomastigotes are left in the bloodstream, which are taken again by some triatomine bug. So this inside the triatomine bug, the trypomastigote is ingested. So in here, it would transform to become epimastigote in the midgut. It would actually multiply. It would multiply in the midgut and transform in the hindgut to become the metacyclic trypomastigote, which is again the infective stage of the definitive host, which is the man. Okay, so just a review. So the man actually acquires Trypanosoma cruzi by the help of the triatomine bug. So when the triatomine bug takes a blood meal, it usually defecates. And when the triatomine bug defecates, it releases the metacyclic trypomastigotes along with its feces. So if there is a contamination of metacyclic trypomastigotes in any skin abrasions, that is the moment wherein the host could actually acquire the infection. So for a metacyclic trypomastigote, which is again the infective stage of the definitive host, which is the man, it would go and multiply into the cells. So it would actually infect the reticuloendothelial cells. It would invade inside the cells and it will transform from metacyclic trypomastigote to a mastigote stage or the tissue stage. So when the cells has a lot of amastigotes, it would actually form a pseudocyst. So this is actually the pseudocyst. This is pseudocyst. So if they multiply and multiply more, eventually the cell will burst. So upon bursting, some of the amastigotes will transform to become trypomastigotes. So these trypomastigotes are subdivided into two forms. We have the broader form and the slender form. The broader form is usually less motile, so mabilin sila sa bloodstream, while the slender form is highly motile. So they have the capacity to invade different tissues, so distal tissues. So that is the slender form. But for the catonabilin, the broader form, the which are actually found in the bloodstream, they could it will be again taken by the vector, which is the tritomine bug. So upon getting the trypomastigotes, so therefore again class, the trypomastigote is the infective stage of the vector, triatomine bug. So upon acquiring the trypomastigote, it would transform to become epimastigote in the midgut. It would multiply to become metacyclic trypomastigote in the hindgut. So, and then the cycle starts again. So, in here, we have clinical presentation of T. cruzi, which is the Chagas disease. So, we have the acute Chagas disease and the chronic Chagas disease. So, for the acute Chagas disease, you already know what acute and chronic is. So from acute, always think that it is sudden, and most of the time it is severe. If it is chronic, that means it is what? It is usually takes a long time. It takes a long time to doesn't have any time frame to see the spectrum of disease. So Right here are some signals of portal of entry. So from figure A and figure B, you could see Romana signal, and figure C and figure D, we have Chagoma. So in the arm circle. So Chagas disease is obviously caused by Trypanosoma cruzi, but the pathogenesis and epidemiology differ greatly from the African trypanosomes. So this is truly a disease of the Americas. So ranging from the southern US, so that is from Texas, so to Mexico to South America. So T. cruzi survives in wild animal reservoirs, such as rodents. So we already know that this is caused by the redovid bug, which is also the kissing bug. So again, the bug feeds on humans while they sleep, and they defecate while it eats, or they after eating they defecate. So that is how these are actually um, transmitted. So, for the clinical presentation for acute Chagas disease, sudden and severe, usually sudden and severe. So, at the site of at the skin site of parasite entry, you could see here in this picture a red area. This develops a chagoma. So most of the patient, if you could remember, the slender form of the trypomastigote has the capacity to invade distal sides of the body. So if it has the capacity to invade the distal side of the body, it has the capacity to do systemic spread. So meaning when you say systemic spread, that characterizes fever, body malaise, swollen lymph nodes. So, swollen lymph nodes, you already know in your anatomy and physiology, that means lymphadenitis, right? So, if there's systemic invasion, there's always fever. That is one way of our body coping up. So, I have read in one journal that if you really have fever, you don't need to take medications. It's just like coping up of your body. So, your body will react to certain pathogens that will enter inside the body. So if your fever ranges from 38.5 to 39, that is the moment that you will take medications. But if it's from 38.1 to 38.4, then there's still no need to take medications. But again, you still need to consult your doctor, of course. But the point is, your body has the capacity to fight these microorganisms, especially the systemic spread of these microorganisms. So that is why you would know later on that most symptoms or signs and symptoms include fever. So there's always high temperature in, that will be involved. So organs that can be infected includes the heart and the CNS, since we are talking about systemic spread. So systemic so tanan. So if you're talking about heart inflammation, so that occurs that results to tachycardia, so meaning the ECG changes. So we have CNS involvement, that is meningoencephalitis. So we have also lymphadenopathy and hepatosplenomegaly. So for your hemoflagellates, it shares almost the same clinical presentation. So it shares the same clinical presentation. So in here, you could read repeatedly hepatosplenomegaly, or the enlargement of the liver and the spleen. Why is that? Because remember, your amastigotes will invade the macrophages, will invade the reticuloendothelial system. So upon invading, there is proliferation of the organ. So the organ will fight back. So there's proliferation of the organ right here. So there is actually um, enlargement of the size of the organ. And in worst scenario, there is destruction of the organ. So in here, hepatosplenomegaly, so enlargement of the liver and the spleen as well. So chagoma, it is very unique characteristic of an acute Chagas disease. So chagoma is a subcutaneous nodule, So subcutaneous nodule, so meaning inside, subcutaneous deeper siya, at the site of the deposition of the bug's feces. So Romania sign, this is what you see in the picture right here. So Romania sign is, it is a unilateral painless edema of the eyelid and conjunctivitis. So this is an example of a Romania signs. So most person will actually um or the most acute most patients suffering from acute illness resolves in about a month. But some patients which enters the intermediary phase, when the patient enters the intermediary phase, in this phase has no symptoms. If you can remember, we have the acute phase, the intermediary phase, and then the chronic phase. So the intermediary phase, in this phase, there are actually no symptoms, but they are persistent, and the level of parasites are persistently low. But most of these patients who are in the intermediary phase will remain in the intermediary phase for life. And for some reasons, some people, some persons who are in the intermediary phase will develop to have a chronic Chagas disease years later. So this is how persistent the parasite is. So even if you already know that most of the parasites are already um, I'm sorry, you cannot really assure that the parasites will be completely eliminated, since most of the some of the patients will experience or will enter the intermediary phase. In this intermediary phase or this intermediate phase, most persons or patients will remain in this phase for life. And if this person or if this patient um, has low immune system, this patient will develop to have chronic Chagas disease, which years to decades later be diagnosed. So again, so we have here the asymptomatic. I think I have already discussed some of this one. So the organs primarily affected are the heart and some hollow organs, and that includes the colon, the esophagus. So this is the clinical presentation of the chronic Chagas disease. But intracellularly, T. cruzi, a mastigotes, cannot usually be found. So, and it is very unclear why disease developed in these organs. So for the heart, usually it would patient suffering from cardiac disease that is caused by Chagas disease would suffer from arrhythmias, so tachycardias as well, and later on there's an increase in heart size on or heart failure. So that is what we call as cardiomyopathy, dilated cardiomyopathy. I think you already know that in your anatomy and physiology. And we have also mega disease. So mega disease of the colon and esophagus. C class, if you could see the pattern, most of the clinical presentation of these parasites could cause enlargement of the organs. So enlargement of the organs could allow or could develop with symptoms of difficulty of um, or pain in swallowing if there is a mega disease of the esophagus, or a mega disease of the colon or regurgitation of the food. So this actually cause still some abdominal pain if the patient is suffering from a gastrointestinal form of disease. So abdominal pain because of an dilated colon or a megacolon. So some patients even can go weeks without bowel movements. So these are some of the clinical presentation of your chronic Chagas disease. So how are we going to diagnose T. cruzi? So T. cruzi, we could still do the peripheral blood microscopy. We could still do the culture. We have antibody and antigen detection and xenodiagnosis. Xenodiagnosis, we have already discussed this in our introduction, way back in the parasitology introduction, you already know that cyno diagnosis involves laboratory fed animals. So they are the fed vectors, rather. So these vectors are being fed with human blood, and their feces will be checked under the microscope. So that is diagnosis. So molecular methods, just remember, molecular methods are the most sensitive, most specific test of all. So how are we going to treat T. cruzi? The doctors will do its job. So these are some of the treatment or drugs that are for T. cruzi. There's no need for to memorize that one. That is not our job as med techs. But what I want you to focus here is this one, the prophylaxis. So how are we going to prevent ourselves from getting this infection? Um, Very self-explanatory. We just have to eliminate the sources of infection, and those are the bugs. So these are the bugs or the vectors that are bringing the infection. So I have placed here, according to the latest WHO data in 2020, deaths in the Philippines reach 2 or 0.0% of total deaths. So meaning T. cruzi is not common. It is not common in the Philippines, but it is still existing, still existing. Next is we have the Trypanosoma brucei complex. So Trypanosoma brucei complex consists of Trypanosoma brucei gambiense, Trypanosoma brucei rhodesiense, and Trypanosoma brucei brucei. So in here, Trypanosoma brucei gambiense is the agent of West African sleeping sickness. Class, I want you to remember this one. This is a very common board exams question. So in here, we have Trypanosoma brucei gambiense causes the West African sleeping sickness. It is pathogenic to man. We have Trypanosoma brucei rhodesiense causes the East African sleeping sickness, which is still pathogenic to man. We have Trypanosoma brucei, which causes the Nagana disease, which is non-pathogenic to man. So therefore, class, we will be focusing our lecture of these two clinically significant parasites. We have the Gambiense, the West African sleeping sickness, Rhodiense, Rhodesiense, the East African sleeping sickness. So for the life cycle, it has actually the same, the same most it has the same life cycle of Trypanosoma cruzi, but it will just have to differ in some certain stages, which we will be discussing later on. But it has the same host. So we have still the definitive hosts, which are the vertebrate hosts and other animals, and we have the intermediate hosts, which are the vector. If your Trypanosoma cruzi is, we have the red ovid bugs. So in here we have the tsetse fly. So we have the tsetse fly. So we have both male and female flies, right? So these serve as the vectors. Take note that in your Trypanosoma brucei gambiense, the vector is Glossina palpalis group. Well, the T. brucei rhodesiense, we have the Glossina morsitans group. So Glossina palpalis group is for Trypanosoma brucei gambiense. For Glossina morsitans group, Trypanosoma brucei rhodesiense. These are Tsetsefly. So the infective form is still metacyclic trypomastigote. It has the same infective form with Trypanosoma cruzi. So the mode of transmission, very common mode of transmission, is the bite of the Tsetsefly. This time it bites the Tsetsefly. So for the life cycle, so this is the life cycle of your Trypanosoma brucei. For Trypanosoma brucei, the bite The mode of transmission commonly is through the bite of tsetse fly. So the bite of tsetse fly harboring the infective stage, which is the metacyclic trypomastigotes. So the metacyclic trypomastigotes will be injected directly into the bloodstream. So it would be injected directly into the bloodstream, which will be carried to the other sites. So the metacyclic trypomasticotes, if you could still remember, there are still there are two forms of trypomasticotes, right? We have the long and then the slender form of trypomastigotes and the short and stumpy form of trypomastigote. Take note that the long and slender trypomastigote is the um, multiplying form, while the short and the stumpy form is the non-dividing form. So from the long slender form trypomastigotes, it would multiply and then it would invade various sites of the body, which includes the blood, the lymph, and the spinal form or the spinal fluid. So, right after invading the bloodstream, take note that your bloodstream again is the superhighway inside our body. So it has the capacity to invade important organs, say for example the heart and the CNS. So the heart and the CNS. So it is usually so the circulating trypomastigotes, meaning the trypomastigotes that are left in the bloodstream, are actually identified through PBS or microscopy. So through PBS or microscopy, PBS means peripheral blood smear. So they are actually be identified and detected using microscopy. So, but um, it is undetectable. It take note that it is undetectable when it is in the latent phase. What is a latent phase again? Latent phase meaning this is the dormant phase. The dormant phase or the inactive phase. Okay, so the dormant phase or the inactive phase. But when the patient is currently on its acute phase, then it is detectable. But when the patient is currently on its dormant phase and inactive phase, don't expect any trypomastigotes seen in your PBS. So these trypomastigotes, which are left in the circulation or in the bloodstream, are then again taken by the vector, which are the Tsetse fly, meaning the infective stage of your Tsetse fly is the trypomastigote. That is the infective stage of your Tsetse fly or the intermediate host. So, from the bloodstream, the trypomastigotes will transform to become procyclic trypomastigotes. So, it will transform to become procyclic trypomastigotes and it would go from the midgut and multiply and transform to become epimastigotes. So, they would multiply and become epimastigotes in the salivary gland. Take note that they will multiply in the salivary gland. That is why in the salivary gland, the epimastigotes will transform to become the metacyclic trypomastigotes. And this metacyclic trypomastigote that is found in the salivary gland of the Tsetse fly will be injected again to the human host. And the cycle begins again. Okay, so just a review. So we'll start again from the infective stage of the human host, and that is the metacyclic trypomastigotes. So from metacyclic trypomastigotes, it would transform, it would invade and multiply and invade the blood bloodstream. It will invade the bloodstream. So upon invading the bloodstream, it would be taken again by the Tsetse fly. But take note again, Your metacyclic trypomastigotes, and specifically your slender form of trypomastigotes, has the capacity to invade distal sites of the organs or the CNS and the heart. So what was left in the bloodstream is taken again by the tsetse fly. So in the tsetse fly, they will get the trypomastigotes. They will get the trypomastigotes, and then it would transform to become the procyclic trypomastigotes, and then transform again to become the epimastigotes. And from the gut, from the gut to the salivary, it would be it would multiply to become metacyclic trypomastigote, and then the cycle begins again. So from metacyclic trypomastigotes to trypomastigotes to procyclic trypomastigotes to epimastigotes, and then metacyclic trypomastigotes again. So for the clinical presentation of your T. brucei, it has the same course of infection. So we have the chronic and then the acute. So for T. brucei complex, it, it is subdivided into two clinically significant genus and species. For Brucei gambiense, it causes chronic course of infection with slow progression. For Rogisense, we have acute course with rapid progression, or early death. So acute is usually sudden and severe. For chronic, slow progression, and that is effective for Gambianse. By the way, Gambianse is the causative agent for west or east. Gabiense, again, is the causative agent for the West African sleeping sickness, while Eurodiscense is the causative agent for the East African sleeping sickness. Please do not forget. So, what we have here is another clinical presentation. So, your trypanosoma brucei complex cause, could cause trypanosomal chancre. So this chancre is subdivided into stages. So based on the severity of disease. So from stage 1 disease, usually it's febrile illness. So we have the winter bottom sign. Winter bottom sign, usually we have pruritus, maculopapular rashes. When you say pruritus, that is itching. We have the rashes and we have edema as well. For the stage 2 disease, this is the sleeping sickness. So in sleeping sickness, there is the invasion of the CNS. Other than the fact that your trypanosoma feeds during nighttime, um, your trypanosoma brucei complex, to be specific, has what we call as somnolence, daytime somnolence, meaning they have sleeping sickness during daytime while being restless and being suffering from insomnia during night. So during nighttime. So this is the most common clinically significant um, sign of a sleeping sickness. So, daytime somnolence, meaning feeling sleepy during daytime and cannot sleep during nighttime, or insomnia during nighttime. So, how are we going to diagnose patients? Again, we have microscopy, we have serial blood sample examination. So, serial meaning we don't just diagnose the patient with the absence of this parasite in just one collection. With just one collection, we need ample collection to detect and to really catch the parasite. So here, aside from the blood sample, we have CSF examination. If you could remember, your parasite has a capacity to invade distal organs, specifically your CSF, specifically your CNS. So We have the CSF, we have the lymph node aspirates, antibodies and antigen tests, culture again, and animal inoculation. So these are the treatments and prophylaxis of T. brucei complex. So the drugs used for treatment of African sleeping sickness are usually suramin and pentamidine. So the doctors have different treatment that is based on the type of disease. So either it could be the West or the East disease. So these is present what is presented to you is a table of the different stages of this disease. But for the elimination of or the prophylaxis of these disease, we could actually control um, like destructions of the insect's habitat. So we need to control the vector, eliminate the reservoir of sources. So in order to control African trypanosomiasis. Again, for prophylaxis, when we hear prophylaxis, we always correlate that with vaccines. I think I have already mentioned this in our introduction that for the formulation of vaccines in clinical parasitology, it would be very challenging for the scientists. Why? Because your parasites has the capacity to mutate. It has different antigenic variation. It has antigenic variations, and it is their structures are much more complex compared to the bacteria or the viruses. So as of now, vaccines are not available as the parasites undergoes these frequent antigenic variations. So in front of you is the comparison between Trypanosoma brucei and Trypanosoma brucei gambiense and Trypanosoma brucei rhodesiense. This is actually found in your Essentials of Medical Parasitology, if not in your Panickers. So can be scan your book in order to this. I believe we have already discussed everything. So from the diseases down to the vectors and down to its clinical presentation. So I guess that's it. So these are the references from my major references or essentials of medical parasitology and Panicker's textbook of medical parasitology. But if you still have time, you can read the Henry's book of Henry's and also read Zabik's medical parasitology if you still have time. But for the main references, essentials of medical parasitology and Panicker's of medical parasitology are sufficient enough. Again, this is to emphasize that you need to read the book. So do not read bullet type notes. So those notes are set for review styles or reinforcements. As for you, you are still second year students, so you need to develop your comprehension and analysis of these certain topics. So you need to read the books and go back to your books. So those bullet type notes are good for review materials and not for learning purposes or for initial learning purposes. Okay, so that's it. So if you have any questions, don't hesitate to ask me during office hours. You can meet me in the office or email me during office hours. Okay? Thank you so much. And this is again Miss Kate, your lecturer, and I will be seeing you around the school. Thank you.